Welcome back to BioInfo Bytes. Today on this explainer, we're diving straight into the microscopic universe inside us to explore how proteins, the actual heavy lifting workhorses of our bodies, communicate with each other. A lot of times, we kind of picture proteins as these solitary little machines. But the reality, it's much more like a massive, dynamic, pulsing cellular social network. By the end of our time together today, you're going to see the human body in a completely different, highly connected light. But before we can even think about hacking or manipulating biology for revolutionary medicine, we have to ask a super fundamental question. How do these microscopic machines actually find and talk to each other to sustain life? I mean, they don't have eyes or ears, yet they coordinate literally everything, from repairing your DNA to fighting off viruses. To figure out this communication, we have to look at the interactome, which is the complete set of molecular protein-protein interactions happening inside a cell. This establishes the absolute core rule of biology. No protein works alone. Seriously. If a protein is a single user, the interactome is the entire global social media platform where every single connection, every friend, and every unfriend dictates the health of the cell.
Here's our roadmap for today's explainer. We'll map the cell's social network, track those interactions, throw co-IP and yeast-2 hybrid into the ring together, hunt down physical interaction hotspots, and finally, talk about drugging the interactome.
Okay, part 1, the cell's social network, mapping the interactions of human life. It is almost impossible to overstate the sheer scale of the human interactome. When researchers map this expansive web using massive databases like Intact, which holds over 300,000 interactions, really clear structures start to emerge from all that chaos. Just like our own social networks, the cell has its influencers. We call these hub proteins. They are highly connected and absolutely vital. Then we have clusters. These are your tight-knit functional friend groups that hang out and get really specific jobs done. And finally, bridge proteins, the essential mutual friends that link completely distinct cellular pathways together. Moving on to part 2, tracking protein interactions, the true detective work of cellular biology. So how do we actually catch these proteins interacting in real time? Well, one brilliant method is the bimolecular fluorescence complementation, or BIFIC assay. It's essentially an ingenious trap. Scientists take a fluorescent protein called Venus and chop it right in half, leaving a Venus N and a Venus C. They attach one half to a protein of interest, like p53, and the other half to a potential partner, like PDED. On their own, completely dark. But if p53 and PDEDs interact in the cell, they physically drag those two halves of the Venus protein together. The fluorescent protein reconstitutes, and boom, it literally glows green under the microscope. Direct visual proof of the interaction. Part 3, co-IP versus yeast-2 hybrid, contrasting views of the protein social network. Now, what happens when our detective tools give us totally different maps? Well, it turns out how you ask the question completely changes the answer. Let's compare the two heavyweight mapping methods. Co-aminoprecipitation, or co-IP, is kind of like an invite-only VIP party. It tends to map functionally similar tight-knit cliques. In network terms, it has higher triadic closure, meaning if protein A knows B and B knows C, A probably knows C too. On the flip side, the yeast-2 hybrid, or Y2H assay, casts a much wider, looser net. It finds a massive amount of connections, but research shows those connections are less functionally enriched. It maps out broad, sometimes fleeting acquaintances rather than deep, functional partnerships. When you actually visualize a co-IP interactome next to a Y2H interactome, the difference is striking. Even when we filter for high-confidence interactions and look at the exact same shared nodes, the co-IP network forms tighter, denser clusters, those functional friend groups we just talked about. The Y2H network, meanwhile, is much more stringy and diffuse. It's an expansive web, for sure, but it's really lacking those tight-knit cliques. The crucial takeaway here is captured perfectly in this finding from an interactome scale comparison study. Co-IP hubs are essential, true biological influencers that highly predict cellular function. But on the other hand, some of the highly connected hubs found in Y2H assays might just be the result of what we call promiscuous binding. Basically, some proteins in Y2H just like to stick to everything randomly, which isn't always biologically meaningful. And that's exactly why researchers have to carefully choose their assay based on whether they want deep functional insight or just broad discovery. Section 4, Finding Interaction Hotspots, the physical binding mechanisms of the cell.
Let's scale all the way down from these massive network graphs to physical atoms. How do two proteins physically lock together? The answer lies in PPI hotspots. These are highly localized, specific regions, sometimes just a few amino acid residues on a protein's surface, that are absolutely crucial for binding. If the interactome is a social network, a PPI hotspot is the actual physical handshake. The proteins themselves might be massive, but often only a tiny, tiny fraction of their surface area is responsible for making that connection.
Looking at the 3D ribbon structure of two CRAD proteins really grounds our theoretical network data in physical reality. When you zoom in on the interface, you can see exactly where this handshake occurs. Take a look at the amino acid glycine 128. It's helping facilitate an incredibly precise hydrogen bond. We're talking just 2.6 angstroms across between the two protein chains. If you were to mutate just that specific hotspot, the connection would likely shatter, and the proteins would unfriend each other instantly. And finally, Section 5, drugging the interactome, moving from understanding to pharmacological manipulation.
Now that we know how to locate these exact physical hotspots, how do we weaponize this knowledge to cure disease? Well, this is the cutting edge of modern pharmacology. The workflow goes something like this. First, you define your strategy. Do you want to build an inhibitor to block a toxic protein handshake, like you might in cancer research? Or do you want to create a molecular glue to force two proteins together? Second, scientists use massive DNA-encoded libraries, or DLs, rapidly screening billions of chemical compounds to find the one that perfectly fits that tiny structural hotspot. And finally, you validate that chemical hit to ensure it modifies the biology exactly as intended.
But what if the protein target doesn't have a neat, tidy structure? What if it's chaotic and disordered? This is where computational biology is completely blowing the doors off traditional medicine. Through de novo design, using advanced AI and computational pipelines from places like the Baker Lab, researchers are now designing completely novel synthetic proteins from scratch. They can literally program a brand new protein to hunt down and bind to even the most intrinsically disordered or shapeless regions of the cellular interactome. We're no longer just discovering drugs, folks. We are custom engineering them. And that leaves us with a truly profound thought to wrap up on. As our maps of the cell's social network become higher and higher resolution, as we map every co-IP cluster, every Y2H connection, and computationally design drugs for every single physical hotspot, what diseases remain truly undruggable? If life is just a network of proteins communicating, we are incredibly close to holding the master switchboard. Discover, analyze, understand. Something to think about until next time. Thanks for joining me on this explainer.